we can interrupt.
Well, thank you very much, as I said before, plus for inviting me. I've been coming here, I think, for most of the meetings. I remember that when we were nearly snowed in Dublin, and there are worse things than being snowed in Dublin. And for, as I said, supporting my trip. Well, everybody's seen this kind of headline from one of the newspapers that I read anyway, so I don't need to get into it. What I'm going to go over is essentially an extract on an international basis of a couple of papers I've written in the past few months. There are two other papers which are under preparation, and I will draw on some data which is also in these publications. The two papers which you're seeing here are freely available to anybody who wants them, as is indeed the full presentation for anybody who wants it. I will show this slide again later on. Matthew has already alluded to the basic economics of plasma fractionation, which has been historically linked to the concept of so-called last liter and first liter economics, fractionation. And I will show by the end of this presentation that, as Matthew said, it is indeed the case that this model is now not applicable. This is something, although not from a direct MRB slide, it's extracted from his data. I do not have to belabor the point. And in fact, I had decided not to put any MRB slides, but I couldn't resist the temptation of putting up this one, which was generously given to me by Patrick last week. And Matthew has already shown you this data, and it is just to accentuate the situation as we know it now. And let me just state, and we were discussing this during lunch, that irrespective of any measures which are taken in relation to ameliorating the situation of supply, some of which I shall propose myself in this talk, there is absolutely no way that the world's plasma supply can ever come close to filling the full demand which is mandated by the current indications, unless very rapid measures in other ways are taken. So I'm first going to go over how I see, this is as usual just my opinion, the pressures on supply are resulting in a shortage, and this is the list of features which I shall go over with. I will first discuss the transformation of so-called latent to actual clinical demand. In the early 2000s, working with Jeff Stonebreaker from the State University of North Carolina, in the hemophilia space in particular, Jeff is well known. He is a decision analyst and has produced a lot of data, still ongoing work in relation to hemophilia. But in the early 2000s, we worked together to assess the situation of so-called latent therapeutic demand for immune globulin. This follows work which he had done in the decade previous to this in relation to how much factor VIII can actually be envisaged as being needed on the basis of usage and epidemiology. This study, the studies which I'm going to describe, done in conjunction with him, applies the same principles to the issue of immunoglobulin. Now, this is, as I said, decision analysis. It is based upon computerized statistics, but it is reasonably easy to understand the basic concepts. Basically, what we're looking at here is the assessment of uncertainty in a situation where many inputs into the environment which we're interested in are still relatively unknown and intrinsically variable. What you see on the left side are the factors which are put into this decision analysis model, which affect the actual demand for immune globulin across a population. And we have done this for two groups of diseases, the primary immune deficiencies and also subsequent work which is not yet fully published but is under preparation in the neurologies. And the first point I seek to make is that the combined latent therapeutic demand for these two groups of indications is round about 250 grams per 1,000 population. Remember, this is just PIDs and neurological indications. This is based upon inputting into the model the best available data of the key parameters, which include the prevalence, i.e. the epidemiology, as well as the dosage and other clinical parameters. So when people look at the figures which are available now for countries such as the United States, Australia and Canada, and see 250 grams per thousand or thereabouts, this is not to be surprised about, because this is in fact a reflection of what is to be expected on just two of the most important indication groups. This is shown from one of the papers, and what this shows is that the latent therapeutic demand for just two primary immune deficiencies in the United States actually exceeds the total consumption of immune globulin in those countries, which was underway when this publication came out in 2014. In other words, the demand for just these two indications in the United States was more than many countries, including most on the continent of Europe, were consuming in total. I hope that this is understood, because the situation since then has obviously shifted, and all these countries are using more. I will skip this slide in order to go on with the other factors. But the first point I seek to make is that what we're seeing in the first instance is that when you project demand based on current concepts of guideline-driven dosage and prevalence, and I emphasize the point of guideline-driven dosage and prevalence, you will get these high levels of consumption for just two of the main indications. I now want to say something about SCIG, and this is the classical representation of the pharmacokinetics of immunoglobulin administered, whether it is IVIG, SCIG, or what they call facilitated SCIG, which is the hyaluronidase facilitated Takeda product. Now, I have assessed as much as I can public information. There's nothing in the stock which is not public information. This is according to one of the analyst groups, which produces these nice reports for which you have to pay $10,000, showing the expected growth of the subcutaneous versus the intravenous market. So it's still projected to be relatively small. But in relation to converting from IVIG to SCIG, and I have some more data on the rates of conversion in a second, there is an aspect which needs to be appreciated, which is that primarily in the United States, because thankfully this didn't happen in the EMA-mandated environment, but primarily in the United States, the FDA, for reasons based upon its assessment of the clinical data submitted to it, introduced this conversion factor of nearly 1.4 in relation to the dosage, which therefore has to be increased. In other words, if you go from IVIG to SCIG and switch on that basis, your switching has to involve an increase in the dosage. And this slide, according to Grifols, indicates that in terms of the PID market, the estimates are that SCIG has actually gripped this market to the extent of 61% in the United States. So this is not insignificant, and it does mean that conversion into SCIG may be a pressure on supply, although I emphasize that this strange construct of conversion is not applicable to Europe. I will come back to this when I talk about possible solutions.
Question? I want a question.
Yeah, okay.
If that is the case, how come that your prediction on the previous slide that the increase in subcutaneous was so small?
That's not my prediction. It is derived from an analyst report, of which you can only, unless you pay untold gold, you can only get some minor graphics. And I am puzzled myself as to why it is in contradiction to the figures presented by Grifols, which are supposed to be current, at least as of 2 years ago. So the answer to the question is, I don't know.
Okay.
And I really do not know what exactly is the situation with SCIG in the United States. I think your community would have a much better idea, and I would appreciate, as I said, I always try to access public information. Now, I want to say something about the question of relative prices, which I think does affect access in certain countries. First of all, it is important to note that relative to other drugs, the price of immune globulin in the past years has not actually increased significantly. It has been more or less stable. Some people might even suspect that it is stagnated. Therefore, there is not an upward increase in price, and the companies have maintained their revenues and their margins by increasing volume primarily, not by increasing price per se in total. But I suspect that that is now changing. This is from an interesting public document produced in Canada, and it shows the IG prices progression in different parts of the world. We all know that in the United States, if you can sell IG in the United States, you're laughing because of the vagaries of the reimbursement system in that country. Companies can expect to get a price from, let's say, 60 to 100% more than they would get, let's say, in this continent. So the price is important. And this is a dictum, my dear friends. The product will always follow the price. This is market reality. The companies, on a day-by-day basis, practically, allocate on the basis of what price they can get. They are obviously constrained by supply agreements, they're constrained by contracts, but they will shift, and I can assure you that this happens, they will shift product from one market to another very efficiently and very quickly on the basis of price. Basically, they will never allocate outside the United States unless they really have a surplus of product in that environment. A shortage in the United States or a tight supply in the United States will affect everybody because product will definitely go there. It doesn't mean that the United States is the only country which gives you a good price. For example, I've heard about Vietnam. It is true that in Vietnam there is actually very little consumption, although it is starting. But the price which the Vietnamese, when they seek to buy, are ready to pay, is quite good. Turkey, if you look at tender data, produces a good price. So does Mexico. You'd be surprised about this, and we can analyze in another meeting as to why this happens. But the product, my dear friends, will follow the price. It is as simple as that. Then there is the question of financial pressures in government reimbursement, and we've already looked at the question of clawback. But I will say something about this, because this has definitely affected the situation in Europe. I know for a fact that this has affected the situation in Italy. Allocation in Italy by all the companies as a result of government clawback measures which have been introduced over the past 2 years has definitely decreased the market availability of commercial immunoglobulin in that country and has therefore stimulated heightened efforts to acquire domestically sourced product, i.e. self-sufficiency. I'm not going to get into the issue of the morality or otherwise the rights or wrong of clawback. You know that the most dramatic example of this in recent years has actually been the situation in Romania. In my previous position, working for a commercial company, I visited Romania a year and a half ago. I was pretty shocked by some things which I observed and were put to me in that country, which I cannot share to the fullest extent here. But this was a very dramatic situation. And just to show you my extraordinary skill in the languages of Europe, I am putting up what was put up in relation to this issue in Romanian ministerial websites. I congratulate the plasma industry for actually somewhat unusually in the context of clawback, because this wasn't the only drug which was subjected to clawback, to actually managing to roll back the measure undertaken to a greater extent by the Romanian authorities. I would say, simply as a final comment on this issue, that I would prefer to see the Romanian government allocate more funds to healthcare in general, because Romania, as a percentage of GDP, allocates relatively modestly to healthcare compared to similar economies such as Hungary and Poland, than to undertake this kind of measure. And then, of course, there is the extremely important issue which we've alluded to, which is the expansion of the indication base. And I'm going to focus on secondary immune deficiency because there have been some recent developments and something which actually we have always known about. Now, this is, as you know, I like to put up data on Australia, not because I'm basically Australian, but because it happens to be very transparent and visible data. So this shows the immense variation between the Australian jurisdictions, which all have fairly similar, in fact, quite extremely similar healthcare systems, compared to the national average. You can see that there is variation. And the national average now is actually higher than what you can see here. It exceeds 250 grams per thousand. And this slide, which I think I had sent to Johan some months ago, shows the breakdown of consumption in the various jurisdictions as well as the national in terms of the main groups. And I have simply pinpointed myself the groups which fall under secondary immunodeficiency over there, which includes the malignancies demanding supplementation by immune globulin, as well as other types of secondary hypogammaglobulinemia subsidized and supported by the National Blood Authority. I reinforce my previous comments to Bob Perry's intervention that Australia is not some kind of Wild West market where anybody can prescribe, where all sorts of off-label indications are prevalent, all these legends, all these fantasies which people speak about when they talk about the usage of immune globulin, because they want to cut down the use. They say, oh, it's all wasted. Everything in Australia is prescribed prospectively on the basis of alignment to national guidelines, which are drawn up by the National Blood Authority through intervention by the key opinion leaders in the usage of the product in the country. And if you don't conform, if you don't align to these indications, you don't get the product. You can get the product by paying, but if you want to be one of the people who gets free immunoglobulin, this is what you have to do. Now, the important thing about this point is that you can see the huge amount which is being absorbed by the secondary immune deficiencies. And I would suspect that the situation in many other countries where the data is not so easily visible to be approaching a similar state of affairs.
Albert, can I just ask a question there?
Yes.
This graph is from 1 year, or?
Yes, it's 1 year. It's from the annual report, but I mean, it's the most recent annual report of the National Blood Authority. I've shown similar graphs to this of previous years. This is the most recent annual report.
So what would interest me is the increase in SID usage over time. Because there has always, what I'm trying to get at is there has always been SID as an indication for immunoglobulins. And the main part of that, the main percentage was CLL and multiple myeloma. This, I don't think, percentage-wise, has changed very much. So what I'm basically interested in, how has this changed over time?
This is the first year that the National Blood Authority has given this breakdown across jurisdictions. And this is actually from a presentation in a conference by one of their people. But I can tell you that, if I'm not mistaken, they've always broken down indications according to, you know, PID, CIDP, secondary immune deficiency, and so on. Up till 2 years ago, the predominant, in terms of the highest percentage consumption, was from CIDP. As of 2 years ago, this has shifted to secondary hypogammaglobulinemia. Now, this is a slide which looks at some experience across Europe in terms of the various hematological malignancies and the extent to which they're treated with immunoglobulin. And in order to save time, I will skim over this slide, but if people wish to discuss it later, that's fine. It is the case that this has always been an indication. Oops, why did this thing rush away from me? Stay there. But in last year, or a year and a half ago, the core SPC was further detailed in relation to dosages. So now we have, after due deliberation by experts, a very concrete evidence-based dosage for these. And this is the proposal from the, I think, process which I think was strongly influenced by Jackie and others from the Croit group, together with Isabella Quinti, my long-standing collaborator in anything IG, and a co-author on the two papers I've shown you before and the two which are scheduled for later. So these are the indications, and this is the situation, therefore, in terms of the recommendations to the core SPC. And this is what happened. So unquestionably now, and I think that I am making this point because, rightly or wrongly, my impression is that secondary immunodeficiency has been only nebulously considered in relation to this issue. Everybody's been focused on the neurological autoimmune disorders, but I think now that we can expect this to be increasingly important. And it is very difficult to model this. I have started work on modeling the actual potential latent therapeutic demand, but it's a bugger. You need to appreciate that you're looking at a diversity of diseases, a diversity of clinical practices. It is verging on the impossible. And in my first simulation, the uncertainties were just too large to produce a credible result. So until the dust settles a bit, and I don't know when that will happen, I suspect it will be impossible to give a strict quantification to this particular issue. I now wish to focus on more important things, which is how are we going to address what I firmly believe is the biggest challenge of this community of plasma users and its attendant industry in the past 30 years, since we overcame the viral safety issues. And I've got these threads of thought, if you like, which are more deeply discussed in the publications, which I would like to go over. First of all, I want to talk about the question of yield. Matthew has emphasized the crucial aspect of yield in manufacture. It is definitely the case that the yield from any production process is one of the most jealously guarded secrets of the plasma industry. It ain't easy to get this data. If you have a reasonable level of expertise, you can actually dive deep into the publications in the public domain of the relevant scientists who developed processes, and you can get a fair estimate. This very public table, which was published by a group of Italian interventionists early last year, is interesting because it compares the yields accruable from what is now an open market for contract manufacture in Italy. So this is public information. And the yields which you can see there on the slide are actually provided in public information, which is why the authors were able to cite them, by the relevant companies in their tender submissions for the business. And these actual submissions are public information. You have to dig deep to find them in the labyrinth of Italian bureaucratic information in the internet. You have to obviously know Italian, but I have assistance in that regard. And you can get the information. But in any case, these guys did a very important service for us by putting it there. And you can see that there is a marked variation in yield. These people put on there as public information the relevant companies. You can see that there is substantial variation in the yields. And this is very important. Now, as an old fractionation chemist, I can tell you that when Edwin Cohn, of blessed memory, developed fractionation, he had not the slightest interest in immunoglobulin. His basic scheme was to extract albumin from plasma and get the best possible yield for that. The Cohn fractionation classical method 6 is not attuned to optimization of Ig yield. A yield of 3.5 to 3.7 is reflective of the Cohn basic unmodified scheme. You can't do better than that. If you want to do better than that, you have to look at the chemistries which started to be developed 20 years ago. First by Bayer, which became Talacris, which became Grifols, and then by CSL and so on. Using so-called caprylate precipitation, abandoning the Cohn scheme reasonably early on. These unquestionably result in a gain of at least 1 gram per liter of plasma. Until we got used to how these fractionations affect other products, these fractionations actually led to unsafe products. It was these fractionations which initially allowed more factor 11A to break through into the product. It was these fractionations which allowed more hemolysis. And until measures were taken to ensure that these adverse events were minimized or avoided, this was the situation. However, now the products have these measures and they definitely allow a higher level of immunoglobulin to be gained. So the first lesson is focus on yield. And if you're in the business of influencing contract manufacture in your relative countries, make sure that whoever gets the job, if there is a choice on the market, is able to deliver the best possible yield. And the best possible yield, which is accruable today by the industry, is in the region of 4.8 to 5 grams per liter. I want to say something now on dosage, and I stand ready to be corrected by Jackie, who wrote a magnificent paper on this some years ago in a journal, in a special edition which I co-edited with Isabella. But I want to speak specifically about the issue of the SCIG versus IVIG correction factor, because real-time data in the United States, now, as I said, this is not a problem for Europe, but the biggest market is the United States. So anytime there is a shift in SCIG to IVIG conversion in the United States, we can expect this to consume more and more immunoglobulin. But real-time data shows that actually you don't need to do this, that you can gain the same clinical outcome in relation to the wellness, if you like, of the patients by maintaining a dosage ratio which is closely approximating to 1, as is indeed required or recommended by the European SPC.
And Albert, that's what I'm told in Canada, that it's actually resulted in a decrease in utilization.
Yes.
If you switch to SCIG.
Yes. So my message on the basis of this is to basically examine closely the situation in the United States and, if possible, generate this type of data through the companies, because it's the company individual applications which in the end have to be amended for this. For example, CSL, in their first application for, I think it's called Hizentra, extraordinary the names of products these days, incidentally, for their extremely highly popular 20% subcutaneous product, were allocated a conversion factor of, I think, 1.45, which the FDA then revised to 1.37 in the second application. So on the basis of data, there's always the possibility of influencing these events. Now, I've already voiced some perhaps exceedingly acerbic remarks about the issue of personalizing treatment, but I'll go over it again. This is a composite slide which is attempting to draw our attention to two opposing schools of posology philosophy in IG. In 2010, Jordan Orange, in a study which was supported by, I think at that time they were still called Baxter, derived through a meta-analysis the concept that basically immunoglobulin, it's a bit like putting petrol in the tank, that the more you put in, the better the clinical outcome. He did this through meta-analysis of many studies. And so he showed through this meta-analysis that if you had higher and higher dosages, you got better and better results in respiratory complication amelioration. In contrast to that, Isabella Quinty again, through a 5-year prospective study in relation to her own cohort, studying actually patients, not meta-analysis, it is, studying her patients, derived the conclusion that this was only true up to a certain extent, and that when trough levels through appropriate dosage exceeded 400 grams per milligram, the clinical situation in terms of pneumonial incidence leveled off. In other words, she is opposing the concept of the petrol tank. And Isabel and I have frequently referred to the gasoline philosophy versus the prospective philosophy. I do not wish to engage into controversies about this, but it is the case that this is allowing us now to start approaching more and more the question of personalized medicine. This is a very famous set of data coming from American investigators from quite some time ago, showing the dangers of allocation of treatment on the basis of third-party insurer-imposed trough level ranges. Sorry about the length of that sentence. I understand it is complex. But basically, in the United States, there was a time, I don't know if this is still the case because I haven't lived there for some time, when the insurers said, okay, we will reimburse your IG if you are within this range of trough level. In order for you to be reimbursed, you have to be in this. But as was shown in these two particular case studies, patients don't all behave the same. And your biological level may well be different from these kinds of guideline-driven, if you like, because these trough levels are based on guidelines which are supposed to be based on evidence. But I'm cautious about guidelines. And biological levels of patients, of course, can vary either way to these trough levels. And for this reason, now, we have different algorithms being proposed. And this is from, again, Isabella, which is an algorithm which she uses in order to actually decide which PID patients are going to get IG. She does not give IG to all PID patients. She studies them in her hospital. This is relatively, I wouldn't say easy to do in Italy, but in Italy, any intravenously administered drug has to be done in a hospital. You can't take IVIG at home. You can argue for this every which way, but this gives Isabella the opportunity to do the work, the magnificent work she's done over the past 20 years, because she studies these patients assiduously. And this is simply, I won't go into the medical details of it, this is simply a way of using laboratory and clinical history parameters of deciding whether people will actually take IG or not, whether they will benefit or not. This is another similar paper from the Quinty group in which she uses similar concepts in adjusting dosages rather than the make or break decision. I won't go into the detail. And just to not give the impression that I'm partisan for the Quinty group in Rome, this is a very nice paper from Steve Joles in the United Kingdom, which in fact proposes pretty much a similar approach in relation to the concept of personalizing. Now, personalized medicine is the sexy thing. Everybody wants to personalize medicine. But I reiterate my previous remarks. Personalizing medicine, to be successful, has to be done intensively and developing very strong links between patient and doctor, involving hospital stays, involving expensive investigation, and it must not be seen, as it is unfortunately seen by many healthcare bureaucrats, as a way of cutting costs. Because in fact, it's probably the opposite if you really exert good medicine. But what are we about? Exerting good medicine or saving money? In relation to IG, we have no choice, because this drug is not going to be available for many people unless this kind of approach is strongly considered. And then there is the question, of course, in relation to personalizing treatment, of the possible role of antibiotics along with immunoglobulin for PID patients, who I think are extremely experienced in getting antibiotics. But I think there is a resurgence now, and this is a clinical study which is not yet, I think, I don't know, Jackie, has this been done, or is I think the paper, this paper is from the Quinty group and a lot of other clinical immunologists in Europe, which proposes a very large trial, a very large study across the continent involving the co-treatment with IG and antibiotics. And I know that the state of the art demands IG for prophylaxis, but I think we really need to look at every possible option. Now, next possible approach, next possible amelioration. Okay, increase and diversify the plasma supply. I think we've said this before. This is the current spectrum of proposals. The increase of the plasma donation rate frequency in the continent of Europe by donors under the, if you like, public health environment. This is an interesting breakdown of the donation frequency, because what this shows is that this discussion as to the frequency, and believe me that this is a very hot discussion at the moment, is really missing the point, to quote Matthew. because the actual approved frequency is never really a realistic reflection or a realistic forecast of what donors will actually donate. Even in the, let's say, war environment of the United States in terms of plasma donation, where the hypothetical donation rate is 104 donations each year due to the FDA mandate, this is never even remotely approached. Perhaps some donors, a very small minority, will approach this, but the vast majority of donors in both the paid donor and the unpaid donor environment will only donate to a small proportion of what is actually approved. And this is definitely the case in Europe. In Australia, for example, where the European guidelines have been mandated for longer than they have been mandated in Europe, I mandated the Council of Europe Guide 20 years ago in Australia, there's a maximum of 32 donations a year, but the actual median frequency of plasmapheresis in Australia, which, as I said, is a very efficient collector of plasma, is 8 donations per year. But this isn't the only issue in my mind, and I want to raise this because this is one of the sets of data which will be in a further publication, which will be submitted in a few months, which looks at this whole question of practice and sufficiency in the continent. This is data produced for us by Peter Turek at the Strasbourg EDQM meeting pretty much a year ago. And this shows us, my dear friends, the most efficient plasma collection system in the world, the Czech system. The Czech Republic, I nearly said Czechoslovakia, I'm an old man. The Czech Republic collects 61 liters per 1,000 inhabitants, and remarkably, this comes not just from a very high rate of source plasma to paid donors, but it also comes from a substantial amount of plasma which comes from whole blood. Now, you might say 61.1 liters per 1,000 inhabitants, and that one is 6.9 liters per 1,000 inhabitants plasma from whole blood. Okay, 13.4 liters of plasma per 1,000 inhabitants comes from blood establishments, from blood community-based blood system through apheresis. So in total, there is 20 liters of plasma per 1,000 inhabitants in the Czech Republic, which are coming from the voluntary system, and about 41, which are coming from the commercial system. So, congratulations to the Czech Republic for attaining self-sufficiency, because if this plasma was all to be fractionated into immune globulin, obviously it would have to be done under contract because the Czech Republic possesses no facility, it would yield, ladies and gentlemen, about 250 grams per 1,000 population, which I remind you is what we estimate is needed on the basis of modeling for 2 of the main indications, pushing aside gently and reluctantly the question of secondary immune deficiency. So you might say the Czech Republic is laughing, but it's not, because the consumption of immune globulin in the Czech Republic is obviously inadequate. Irrespective of how you measure the justification of demand, it is clear that Czech patients are not being given sufficient amount of IG, and this is why I would maintain that the core issue has always to come back to the clinical environment in respective countries, whether there is over or under usage. Nobody on God's green earth can suggest that the Czech Republic has overusage. It has underusage at the same time as providing the world's highest rate of plasma for what is essentially commercial endeavor. And you can discuss this in relation to the pluses and minuses later on. I think from my tone, you can perhaps assess my own opinion. Possible solutions have to include pursuing alternatives. And this is particularly accessible, in my view, for the autoimmune pathologies, because for any kind of deficiency of IG, whether it is congenital or acquired through hematological malignancy, has to result in substitution of IG. But the whole question of the autoimmune pathologies, whether you're looking at the parent indication, ITP, which we've already mentioned, or the whole question of the various indications which absorb much more. Now, ITP is basically irrelevant really these days, although it still underpins a lot of regulatory thinking. It has a huge consumption. Now, the reality is that these pathologies all have alternative treatments. The alternative treatments may be infinitely less convenient. They may even be less safe. Steroids are not nice things to have. There are adverse events. They may even be considerably more expensive. Putting a person inside a plasmapheresis unit and subjecting them to plasma exchange isn't just an undesirable thing for the person, but it's also quite expensive. But they exist. They exist. This is the Cochrane Review in relation to that. This is a systematic analysis, a systematic review, a meta-analysis of the trials of rituximab, which is a monoclonal antibody now very long-standing, used. It's an anti-CD20 and it's used in a variety of disorders. It's also used in a variety of blood cancers. And it shows that the meta-analysis indicates a benefit of this disease, of this modality for this disease. So this is something which can be considered. And this is a pooled response from the same study for each disease subgroup from the observational studies. So we already have one drug, not cheap. I keep emphasizing, this is a question of economics. Although rituximab has now been, if I can use a word, biosimilarized, because the patents run out, it's been biosimilarized by people who are in big pharma. And so they're making still a lot of money from it. It ain't cheap to treat a person with rituximab. I don't know how it pans out relative to IVIG, but it ain't cheap. But it's there, and it should be investigated even further. These are, in relation to Alain's previous question earlier this morning, this is a synthesis of these next-generation biologics currently under investigation. Very commendably, very intriguingly, the three main plasma companies, which, as has been mentioned earlier on, account for basically more than 2/3 of the world market, have all purchased intellectual property into these technologies, and they've actually purchased the companies themselves, the startups which involve these technologies. And the question is, so what's going to happen? My personal view is there is some way to go, but I think people like Brian and David and Mark Skinner will agree that 12 years ago we had to plunk into PubMed the word emicizumab, longer-acting factor 9. I think all these things will not have popped up. I think that if you pop these things into the PubMed system now, they do pop up because there are already studies. And my personal experience tells me that where there is a real medical need, science will accelerate its efforts to fill that real medical need. And I trust I make myself obscure. This is showing the very basic reality behind this. This is from some years ago, and I think this is the United Kingdom situation, but it is pretty much a reflection of what is the situation overall. And if these modalities, which are primarily directed towards illnesses such as CIDP and Guillain-Barré, and even ITP and myasthenia, and all these autoimmune-directed pathologies, result in alternatives, this is going to have the most profound effect imaginable on the economics, and I told you, I'd show you again the slide, of the plasma industry. This slide is from a book which is now venerable. Anything older than 5 years in this business is venerable. That's why I'm ultra venerable in this business. I am very venerable, as well as being very vulnerable. But you know about the concept of first liter and last liter economics and the need to extract 3 proteins to be viable. This has already been dead and buried. Matthew is quite right. Because of the inability to sell plasma-derived factor VIII, the reality is that the bread and butter are coming from IG and albumin. Albumin is really very important. Because IG, some estimates, I tend to agree broadly with them, IG, if you sell IG in the United States, you will cover your plasma purchase and your manufacturing costs and make a wee bit of margin. But then you have to sell more. And this is why the companies who are wealthiest because they get more revenue per liter. Besides selling albumin, a huge amount of it in China, they also have high-value proteins such as alpha-1 antitrypsin, C1 esterase inhibitors, and intelligent forays into markets such as for fibrinogen and the old APCC product, which are gaining them very useful revenue, a good return on investment. Well, but it's still bread and butter. The bread and butter of immune globulin depends enormously on one single disease, CIDP. If that disease is able to be covered by an alternative modality, the effect on the industry's economics will be substantial. Another issue which is extremely important, and I'd value Matthew's view on this, is albumin in China. The companies sell albumin in China, and it is extremely important, not just as a revenue flow, but because of the particular aspect of China, which consumes albumin at a very brisk rate. Actually, over the whole country, China is a median consumer of albumin, but the health system in China, its relevance is based on the big cities of the developed part of China. These hospitals consume albumin inordinately. I was last in China a year and a half ago, and I had an interesting meeting with what I would call a key opinion lever puller in China. And he was explaining to me the whole situation of why at that time albumin in China, usage of albumin in China had gone down. It was because of the direct intervention which a command economy makes possible. The government can intervene at any time to influence clinical patterns, and at that time it did. When it saw that its domestic fractionators still were not able to meet the demand, it realized that it had to be a bit more circumspect. But I am, for better and for worse, I may well be wrong, I've been known to be wrong, I am confident that the albumin market in China is not something which the companies can aspire to hold on for ever and a day. I think that this market will again come under pressure, and when that happens, there's going to be substantial implications, because albumin sales in the United States and in Europe are nowhere as robust, especially on this continent. So keep in mind that the economics of the industry is becoming more and more focused on one product, and this product is more and more focused on one or few indications, which are exerting their influence on the current situation of supply and demand. Keep in mind that this is a commercial industry and is driven primarily by the need to return an adequate profit to its shareholders. These are the people who the companies are answerable to, and they must deliver. And so, how are we going to adopt some smarter way of actually getting these products? Well, again, prices, not easy to get prices. I mean, some companies, some of these investment companies, analysts, they sell you these reports and they claim all these prices. I got these, all of them, from the various issues of the International Blood and Plasma News, which is one of the most useful resources in this industry. I'm intrigued by these prices. Not easy to get these prices, but these are from IBPN. Well, I'm just intrigued that the national fractionator in the Netherlands charges 71 euros per gram. I'm intrigued by the prices which are available to the Japanese Blood Product Organization, 67 per gram, compared to the imported price of 45 per gram. And I continue to be awestruck, not intrigued because I know why, but awestruck by what is achieved by the Australian government, which pays 36 euros per gram for the domestic product and 28 euros per gram for two products which should be familiar to you in the global environment and the continent of Europe. Now, hands up those countries in Europe which are paying 28 euro per gram for Privigen. Flebigama? No one? Of course not. Now, you might say, well, they love Australia. It's such a nice country, and all these poor kangaroos have been roasted. So they're selling it cheap. I don't think that they're selling it cheap. I think that this is the market. This is a tender system. In front of me is the world's #1 expert in how to manage tender systems in this space. Remember this, these products are all available. They're all freely disbursed. The policy is not to have one product, not even to have two products, but for security of supply to have at least 3 products. All these have a presence in the market. Obviously, far and away the biggest presence is the domestic product, but that has 50% of the market, that one product. The other 2 share a reasonable slice of the action between them. And you might say, so how does the MBA manage to acquire these interesting prices? Well, I can summarize it because in my previous incarnation, but 2, I was an advisor for the National Blood Authority, and they achieve it by looking people in the eye and waiting for them to blink first. And I trust I make myself obscure. So if I can round off, and thank you very much, Jackie, for your, as usual, infinite patience. The industry is increasingly dependent on immune globulin sales. There are other valuable proteins. The search for valuable proteins goes on. Probably there are even more valuable proteins. And probably the current proteins, which are not such a high level of demand, can be further exploited in their current indication of congenital deficiency into other acquired deficiencies. But this will take time. And as alternatives to immunoglobulin are developed, my personal view is that they will be, prices will have to come under upward pressure. We need to coordinate efforts to increase plasma supply, and we need to really focus on the issue. I remain frustrated at the amount of energy which is consumed on issues which are relatively unimportant for patients. I am a patient myself. I think that the industry and the healthcare system exists to serve me as a patient and taxpayer. And we need to therefore coordinate and not waste energy on irrelevancies. However, there's also the public interest, and this needs to balance the commercial realities. If the public interest is able to generate prices through intelligent tendering, which can drive down the price, then good on the public interest, and the commercial interest can attempt to gain its interests in this kind of dynamic. And I've already made a plea for personalized therapy, but this needs to overcome guideline-based treatment. Guideline-based treatment, so-called evidence-based medicine, has served us well. It was an advance on anecdotal medicine, which is what we had before McMaster University intervened. And now we are in an era where, thanks to better capacity to assess medical care, better capacity to assess genomic issues, and so on, we can personalize treatment better. But I emphasize that this has to be done from the point of view of best medicine rather than cheapest medicine. And that's all I have to say for now.